I will go through some of this a little bit quickly, and the rest of my time I'll just steal from Clay. If you are really desperate for some good science on your drive back, like Scott mentioned earlier, a longer version of this is available on the website, or if you need help falling to sleep when you get home. So anyway, I'll jump right in here. I don't have to tell anyone in this room that the transition to lactation period is a period that is full of a lot of challenges, but also one that's full of opportunities for us as nutritionists and as managers. A lot of this is centered around the onset of negative nutrient balance, and if we look at the prepartum period, cows are consuming, here in yellow, more than their requirement in blue, and so they're in positive energy balance. And we know that the dry cow, the pre-fresh cow, is a little bit of an easy keeper, sometimes too easy of a keeper, but that all changes very quickly as she approaches parturition and we have the onset of lactation, where her requirement drastically increases and what she consumes can no longer meet that requirement, and thus she enters into negative nutrient balance. We talk a lot about this being negative energy balance, but as Mike pointed out first thing this afternoon, this is more than negative energy balance. This is negative nutrient balance, okay? It's negative macronutrient and negative micronutrient balance. And so one of the things that we focus on in my group, and you've heard from other groups as well, is looking for nutrients that can modulate these pathways to either meet needs directly or can help modulate the pathways to improve nutrient use efficiency or to improve metabolism so that the nutrients can be used more effectively. So what I'll share today is one of those nutrients. You've heard others talking about it as well, but we've been looking at rumen-protected choline. We'll talk about the impact on lactation performance, a little bit about how we think this is working in the transition period, and then also what the impact is, and you heard Barry talk about this just a little at the end of his talk, but how we think supplementing choline in the cow is impacting the offspring's growth and health. So Jose did a great job setting this up, and Usman Arshad is in the back of the room here, the first author of this meta-analysis. So I don't have to go through this, but it really nicely demonstrates that across the breadth of literature, across different products, different feeding ranges, we see a positive response to rumen-protected choline. But the other thing that this study pointed out to us is that if we look at the far right side of these figures, where we would be focusing on higher supplementation rates of rumen-protected choline, we don't see a lot of studies. So we wanted to do a study that would compare maybe one of these higher doses or higher supplementation rates. So we did a study with about 116 cows enrolled before calving, 21 days before calving, and we fed them in these electronic feed gates prepartum where we can assign cows to different gates. We get everything about their intake, their feeding behavior, their meal patterns, and their individual intake. And in this, we had a control treatment, just as one of the studies you heard earlier, we controlled for the non-choline parts, so the lipid protective moieties. And then we had an RPC1, this is the recommended dose that a lot of you are probably feeding or hear about, and this was of a Reassure product. RPC2, okay, is the recommended dose of a higher concentrated prototype. This just allows us to free up space in the diet so that we could feed more of it in our last treatment, which was RPC2 high dose. So these treatments, I really want to point out, these treatments were fed prepartum. We wanted to ask the question, what would happen if we fed more choline prepartum, okay? So because we were focusing on that aspect, when the cows calved, they went on to some really consistent postpartum diets, basically the default of what we would normally feed. So control cows stayed control, RPC1 regular dose stayed RPC1 regular dose, RPC2 stayed RPC2. But that high dose came down to be the recommended dose postpartum. So again, we're only asking the question, what happens if we feed more choline prepartum, okay? And then once those cows hit 21 days, we rolled them into lactating cow diets where they came off of their treatment. We mixed them all up in pens of 16. We followed them out much longer into their lactation. So this is what happened when we looked at, I'm only showing energy-corrected milk here for the sake of time, but we have the supplementation period, so 21 days prepartum to 21 days postpartum. These colors are going to be consistent throughout. So control is red, RPC 1 regular dose, this yellow or gold color, RPC 2 regular dose green, and RPC 2 high dose blue. And what we observed during the supplementation period is no difference. And that is not what we expected to see, because every other study told us we should see a response in the first 21 days. But what we did notice, and I'll put this in perspective for you in just a minute, but what we did notice is that these cows are basically a very steep slope of production that whole first 3 weeks. Okay, so they are increasing in milk really rapidly. In fact, when they get to the post-supplementation period, these cows were averaging in the 120 pounds of milk a day right away. Okay, and so we think the lack of response in the supplementation period was just a lack of time to get to the higher milk production, because we do then in fact see the increased milk, energy-corrected milk, tendency for increased energy-corrected milk in the post-supplementation period all the way out through the carryover period. And that was about 5 pounds of milk a day if we just pick out 2 treatments, the control and the RPC2 regular dose here. Okay? And this is consistent with what we observed in the meta-analysis. What's really interesting here, and I said I'd give you some perspective, is that we've taken these meta-analysis figures and we've drawn or overlaid the milk yield and the energy-corrected milk from the cows on our study. And you can see that during the supplementation period and during the post-supplementation period, the cows in the study were making about 30 to 35% more milk and energy-corrected milk than the average across all the other studies in the literature. Okay, so even in very high-producing cows, we observe the same 5 pounds of milk a day that we observe when we look at all of them taken together. And so that's really interesting to me, because people always want to ask, well, it's easy to get 5 more pounds of milk if cows are only making 60 pounds. Will I still get that much more milk if the cows are making 120 pounds? And so in this study, I think that it demonstrates we will in this case. All right, so how's this happening? And you heard Barry talk a little bit about mechanism and some immune response. How is choline affecting production during, and maybe more interestingly, after supplementation is over? And you heard a little bit about this from Jose. We used to just classically say it's because of liver lipids, right? It mitigated lipids. And there's been some nice work on this, and I won't go through this because Jose already did. But I will say we can mimic this in cell culture. So if we isolate primary hepatocytes and expose them to fatty acids, we can get the cells to do the same thing that the cow does, where the cell will load fatty acids and store them. And that gives us a really useful model to study this, since we don't have the confounders of dry matter intake, retained placenta, metritis, mastitis, all of these other things, right? So we can dig into mechanism in a really focused manner. So over the course of several series of experiments, a PhD student's entire time and many years of some others, we did a series of experiments where we looked at the mechanistic pathways that choline was influencing. We see an increase in the ability of the cell to make energy from fatty acids. We see a decrease in the conversion or the secretion of ketone bodies. We see a decrease in triglyceride accumulation, decrease in oxidative stress, and we see an increase in glycogen, even in the cells. So this was some of the early work that demonstrated to us that choline is acting through all of these different pathways. So that the choline is acting through more than just lipid accumulation, okay? The other way that choline is having activity in the cell is as a methyl donor. So there's a lot of methyl donors that we have access to in nutrition. This is just a labile methyl group, so it's a carbon with 3 hydrogens. But the unique thing is it has to be donatable, okay? So as a nutrient, to be a methyl donor, you have to be able to get rid of the methyl group, okay? And only some nutrients can do that. So methionine has a methyl group here and is a methyl donor. Choline has 3, as does its downstream product betaine. and folate, the one we talk about the most in human health, has 1. So what we know about methyl donors across species and across the literature is that if we have a lack of them, if we have a deficit, we have increased liver inflammation, decreased liver oxidation, and decreased DNA methylation, which we know is especially important in the developing fetus. So anytime we're talking about a lactating cow, for the better part of her lactation, she's pregnant, right? This is something that should be piquing our interest as nutritionists, and we should be asking, what does this deficit or supplementation during this period do to the developing calf? So we have some data out of Florida, Jose's lab, that looked at this across several studies. They found an increase in average daily gain, about 1/10 of a pound per day in heifers, about 1/4 pound a day in bull calves that were exposed to choline in utero, and a reduction in fevers. And the first study from this was with Charlie Staples, and him and I sat and talked about this. And talked about fevers and pneumonia and things like that, really getting at immune maturation probably being increased in those calves, and an improvement in lung development and maturation. So in our study, we wanted to look at the offspring from the cows that we had. Now, of course, we had female Holstein calves, but just like many farms, we are breeding the bottom half of our genetics to beef semen. And so we have male and female Angus Holstein cross calves. We followed them as well. So if you're starting to gather, this was one of the largest studies we'd ever done, and it was definitely a handful. So we followed those calves out. Before we talk about calf growth, I'll comment on colostrum yield. Barry mentioned that also. So these are our maternal treatments, and you can see that the choline treatments had increased colostrum yield compared to control, okay? And there was only a slight reduction in the BRICS, or the quality, one of the measures of quality, and that was only in the RPC2 high dose. So this is not just a product of dilution. In the 2 regular doses of RPC, we had increased yield by a couple of pounds and maintained quality. So in our study, calves received colostrum from a cow within the same treatment group, not necessarily her dam, but the same treatment group. And we measured it, so we only gave them the same amount. So I'm showing you this as a response, but this is not responsible for the difference in the growth as far as volume. All the calves got the same amount of colostrum, regardless of how much the dam made. So when we looked at the growth response, all calves were born without a difference in body weight. We fed a step-up milk replacer program where they received the same amount 1 to 2 weeks, and then an increased amount 3 to 8 weeks. And in the Holstein calves, we observed a tendency for improvement in calves in the first 2 weeks, which was really interesting. But even more interestingly, challenges happen on farm, even when you're in the middle of a research study. How many people have felt and faced really challenging times in the calf pad? Like calves get sick and they bloat or they have pneumonia. And I'll tell you what, for a long time, a lot of us doing transition cow research ignored calves. I certainly did. I'm guilty. I just did the cows and I left the calves alone. And in this study, we did calves, and I have never faced something so frustrating as sick calves, because you want nothing more than for the calves to not be sick, right? So challenges like this happen even to us during research projects. So first, I'm going to start off by saying there was no difference in bloat, respiratory, or fecal scores by treatment. So this was not a treatment thing. We had calves randomly get bloat across all treatments, and there was no interaction of maternal treatment and bloat on average daily gain. Okay, so this is a retrospective side thing we did to try to learn something from a bad coincidence that happened. So what we had was we had some bloat that occurred in about 30% of the calves on the study. It was really cold when calves started hitting the ground, and we were feeding milk replacer, which is not our norm on farm. And one of the things that we did, because we were already taking blood samples, is we looked at serum LBP. Okay, this is LPS binding protein. You heard about LPS already. So LBP is a marker that can tell us basically if the gut integrity is in place. So if LBP gets from the gut into the blood, then that might suggest that the gut integrity is sacrificed. So as we would expect, these are all the control calves. The control calves that didn't have bloat, the lighter bar, had less LBP in the serum than the ones that did. That makes sense. Okay, that doesn't surprise us. What was really interesting to see was across all of the treatment, all of the calves that were maternal choline exposure, we did not see that increase in LBP in the calves that were that had bloat, any level of bloat. So this response was mitigated. Okay, so this, like I said, is retrospective analysis. We want to follow up on this. I gave a talk and I said that, and our farm manager was in the audience, and I had to assure him, not yet. We're not going to follow up on it yet. We're not inducing it yet. But it is really interesting and something we want to look at. Okay. All right, so the beef cross calves were a little bit different. Where there's an interaction of male and female, I've split it out. The birth weight, again, no difference by treatment, and no difference by treatment in the first 2 weeks, similar kind of step-up program. But we did see an improvement in average daily gain in the 3 to 8 week period in the male calves of the cross animals. All right, so our original comment was that choline would improve DNA methylation, or that might be one of the mechanisms. Did it happen? Yes, in the male Angus by Holstein cross animals, we saw improved global methylation. But interestingly, we saw improved growth in all of the offspring at different times. So what else was happening? There were a lot of differences in energy growth and gut integrity markers. And for the sake of time, I don't have them all here. But both of these papers were just accepted, so we can dig into that more at another time. There's one last set of data I want to show here. And this is centered around the fact that we didn't stop when the calves were weaned. With the beef cross animals, we actually followed them all the way out to processing, okay? So sometimes in research, we do things because it's the primary objective and it's what you set out to do, right? And sometimes you do it because baseline data is important, and we have an awful lot of these beef cross calves around. We don't have an awful lot of growth metric data yet. And the other thing is sometimes you do these things because you're motivated by the desire to bring things full circle. So a lesser known fact about me is that my master's at Purdue was actually with pigs and was with carcass quality and meat science. And so I saw this as an opportunity to go full circle and follow these animals out to processing. Okay, so we followed them all the way out. We put them on a feed efficiency study in their finishing phase. And then at processing, we looked at carcass quality. So the growth data here, this is the weight from 2 to 9 months over that period of time, of course, times in the model. We saw an improvement, a linear dose response with maternal supplementation of choline. Calves were never fed choline, okay? So this is something that was done 10 months earlier to the mom, and we see an improvement in growth and body weight here in the beef cross animals. There was also increased wither and hip height, a linear decrease in plasma insulin, and a tendency for a decrease in glucose. And some of this suggested to us there might be some impacts on carcass quality. So when we finished these animals out, about 700 kilos, 640 for the females, we graded them, okay, except for one control cow that did not read the instructions. They all graded prime and choice, which was good to see. But what was really interesting was that we saw a significant improvement in marbling score in the animals that were from a choline supplemented dam. This is really interesting because nutritionally, there are not ways to improve marbling. There are ways to run it, right? We can destroy it really quickly, and there are genetic ways to improve marbling, but nutritionally, we don't have tools to improve marbling. So this is really interesting. At this point, there's only about 10 animals per treatment because we have male and female splits. It was only half of the offspring to begin with. So this is something that we'd like to follow out and do again. All right, so to wrap this up, again, I know I'm standing between us, food, and beverages. Strategic nutritional interventions during the transition period can have long-term impacts on the cow and on the calf, okay? Mechanism of RP choline action is through improved liver function. We've shown you several different ways. And supplementation during the transition period tended to increase energy-corrected milk, even at the high production levels that we observed in our study. Supplementation of cows with RP choline also improved immune function, metabolic health, and supported the carcass quality in beef on dairy cross calves. And higher supplementation rates, that high dose, supported even further improvements in calves, but not in cows. Okay, so this is where we're seeing some interesting splits. And if I could take the liberty of wrapping up all of the talks, my colleagues and mine, I think some of the things that are really consistent across all of our studies in the last couple of years is that there's a consistent postpartum production benefit that we observe, even in high-producing cows, even in cows with high genetic merit, regardless of body condition score. And there are some mechanisms here that we've thrown out today. I think there's a lot involved in shifting metabolism and nutrient partitioning. There seems to be improvements in health and probably some interaction with the immune function. There's also in utero programming that provides benefit to the calf, where we see benefits on calf growth and health. We see improved feed efficiency to finish in the Angus by Holstein cross. And the mechanism for this, we certainly see increased colostrum yield, although we control for that in our research. We see changes in metabolites and changes likely in nutrient partitioning there as well. So with that, I certainly can't do these studies alone, so there's a lot of people to thank. The key members that led the study were Dr. Henry Holdorf, now with Purina Nutrition, Dr. Billy Brown, now an assistant professor at Kansas State, and many others. These large studies take a lot of hands. And I will point out that Sarah Johnson, one of the key undergrads in this, is here with me at Tri-State. So if you see her, make sure to ask good questions.